Okay. So last time we talked about homework 7, kind of like the state of the art in structural brain mapping. We look at the gene profiling, the gene expressions and all the different parts of the brain. And we found the relationship between the bold signal and the neural activity. Right? And this is through the convolution. And we call it convolution through the hemodynamic response function. So look at the neural activity, you convolute hemodynamic response function, also called HRF, and you get the expected bold response. You add noise to it, and that's it. And we also discussed how Nancy can measure, discovered what is called the fusiform face area, right? So today we are going to look at protocol design, and we are going to discuss this paper, Basic Principles in Study Design. And if time permits, we'll spend some time on linear time invariant systems and convolution. Okay? It's an important lecture. We're going to look at how we design the fMRI protocol. Typically, in the conventional sense. Okay? There are more advanced protocol design issues that we can discuss later. There are new types of protocols that are coming up. But this is, focused on what is called the general linear model analysis. You can also call it GLM, right? Analysis. Okay, so here is the ungraded homework. There is a very nice primer. You can read it full, but for now just do chapter 1. Brain Facts 2018 primer. It's a 70-page document. Very nicely it gives a very high-level introduction to various issues in the neuroscience side. So it has about 10-12 chapters. So just go through chapter 1 for now. I've also given a reference on neuroscience. So there's a book by Kendall on principles of neuroscience. So that's a very good book, very thorough. It's a huge book. And, you can go through, whatever chapters you feel like. It's quite comprehensive. It starts with the cellular biology all the way to the brain mapping as well. It looks at various systems and all. And I also posted a reference on convolution in LTI systems. So you can look at that on Moodle. And this you can take a picture of. There is a course called Medical Neuroscience in Coursera by Leonard White.
It was not accessible.
Huh?
It is not accessible. We would, like, in the past, we've been able to audit it, but now Coursera it does not allow us to audit it for free. We have to buy the course.
Oh, you have to buy the course? Yes. It's no longer
free for us.
Oh, it was running for free for quite some time. I see.
I searched up everywhere, could not find it.
Okay, so then we scrap this, okay? I'll look for another resource. Okay, so then scrap it, okay? Fine. Okay, fine. Before we start project discussion, we are supposed to be doing this project in teams of 2, preferably. And an ideal team would be, maybe you can go up to 3 also. Okay? An ideal team would be an interdisciplinary team. But I understand it's kind of hard to work with somebody unknown to you. Interdisciplinary team by meaning one data science person and one cognitive science person. But in case it is hard, you want to pair up with somebody in your batch or somebody your friend, that's also okay. Okay? And I'll update the topic list and post it on Moodle. But it will not go through much changes. I might add a few projects, take away a few. But you don't have to go through. These are just a suggestive topics, okay? So you can look at it, you can either pick any of those, or if you want to do something on your own, you're most welcome, right? So you can get inspired by those topics, and then whatever you want to work on, you can select. But decide your themes and topics by next week. I'll float a Google sheet for themes and topics, and you can enter your names and entry numbers and the topics. Okay. There will be a midterm presentation carrying a good fraction of marks for your project. What is expected in a midterm presentation? You will define your research problem very clearly. Okay? You would have completed a literature review, what people have done to solve that problem. It has to be, try to make it a research problem. Don't try to do something which somebody has already done. There is no point doing that, right? You want to do something new. So, try to do it like that. You have to finalize the data that you will be working with, right? This is very important because there is a lot of open source data, fMRI data available for free downloads. So you finalize the data. And what we expect is, in addition to the research problem and literature review, you will present some results with preliminary data analysis. So you will download the data, you will do the analysis. And what is the date of presentation? Do you know? Mid-semester presentation. It's typically around the mid-semester examination, right? A few weeks here and there. So you have time for that, it's not. But you should start to plan for that right now. Okay? And your next assignment will get you a little more familiar with the fMRI data and how to analyze the data. Okay? Questions or comments? No? Okay. So, we looked at...
Sir,
yeah.
any quizzes coming up?
There are no quizzes here. There are no quizzes in the course. Should we do quizzes?
No.
I see. Maybe next time I'll do. Okay. So we talked about the bold response, and this is the time when the stimulus is presented, and you can think of stimulus presentation like this, like a square wave, but your actual data will look something like this in the ideal world, but then this will be added noise. Okay. So we talked about how you go from stimulus to response. And the GLM analysis, the main purpose is to find activations. Okay. We already talked about distributed versus localized function. Right now the jury is mostly saying our brain function is highly localized, at least as of today. And now we want to find different localizations, right? More and more areas and more specialized functions. So we have to understand, we talked about this, area X is responsive to task Y. Is slightly different from area X is selectively responsive to task Y. Or you can say area X is more responsive to task Y as compared to task Z. And versus causal connection, right? Causally involved in task Y. So we'll see how to answer each of these questions using fMRI protocol design. And then you can also talk about population study, right? In this group. Area X is selectively responsive to task Y in this group. And area X is more responsive to task Y for patients with this illness as compared to healthy subjects. So all these are the kind of questions we are going to be able to answer once we understand the statistical methodology behind this. Okay, so what is experiment design, or what is this fMRI protocol design? You think of conducting experiments to answer a specific question. You may have start with some research question, and depending upon your research question, you will decide to choose the subjects, and you will scan them in a fMRI scanner. When you are scanning them, you have to present some stimulus, and then get some response. And what is the order, how you are going to present the stimulus, what stimulus you are going to present, all of that actually constitutes the experiment design. Okay? But why do we need to do anything special about experiment design? Why can't we just simply, why do we need to give too much thought to experiment design? Anybody has any comments?
Because the experimental design will decide the kind of inferences we can make about our results.
It's the other way. The kind of inferences you are interested in making, you will design your experiment basis that. Okay? But, yeah, so you need little more detail, little more detail. Anybody would want to share? So think about, just forget about this slide. Just think about a research question. What kind of research question you might want to answer? Imagine you got an access to fMRI scanner and you got a lot of helpful friends and you want to learn about brain function. What kind of questions would you want to ask?
Maybe kind of related to depression.
Okay.
Post-trauma.
Okay, so pick one. Let's see. Let's pick one.
Sorry?
Pick one.
Yeah.
So, let's say somebody gets a trauma, seeing some kind of image or something.
Okay, so let's pick trauma. Okay, so you want to work on trauma. You want to find out something about trauma, right? How the trauma is represented. So then what's your research question? What will be your research question? What is it that you want to study?
What?
Brain region gets elevated or
Okay. Which
region of the brain gets.
region of the brain gets activated?
During
When?
When presented with traumatic stimuli.
What part of the brain connects what we are seeing currently to the past memories?
Which region of the brain gets activated in a person with traumatic, in a person with trauma when presented with a traumatic stimuli? And you were saying what?
What part of the brain connects what we are seeing currently to the experiences we have had before?
Okay. Connects what they are seeing now with the experiences they had before. Now this word connects is a bit vague. Can be a little bit more specific.
Or
Gets that.
another thing that could be like how the connectivity among different bone signals varies with certain event.
How the connectivity, how the connectivity between different brain regions changes with different type of stimuli
Different kinds of stimuli or different.
or with traumatic stimuli. Okay, maybe we can ask that question. What else?
So which region gets selectively activated?
You can say, gets selectively activated.
But that would be assuming there would be a region
That will be assuming that there is a region that gets selectively activated. But if there is, then maybe that's an easy cure for trauma, right? You just cut that region off, That's how the neuroscientists would do. Or you sort of, at least, design drugs that target that region. So this is about connectivity studies, right? This is about, when you talk about connectivity, you're looking at functional connectivity. I briefly talked about, right? How two different regions, the bold stimuli is correlated. And that, I think, we can skip for now, because, what we are doing right now is just activation studies, right, for the whole GLM analysis. So if we stick to this, the first one, then let's try to refine it a little bit more. I can see a few loopholes. So the traumatic stimuli that you present, is that a subject-specific stimuli or is it a general stimuli?
So,
Subject-specific, right? So different people will have different traumatic stimuli. Now, which region of the brain gets activated? You want to decipher it for one subject only, or you want to decipher it for a population? Are there some regions that are involved in all the people with trauma?
maybe we should differentiate.
I've read the paper where trauma patients, those who suffered accidents, they were shown pictures of accidents versus some other trauma-related pictures, and they responded more to the car accidents and other accident pictures. So maybe you could take patients like that and categorize them, and then show them such pictures.
Maybe we pick up the most common traumatic experiences that people have, one of the most common, so that it will be easier to get the data, and then get samples of people who have faced that specific traumatic incident, and then provide that as stimuli, mostly visual, I would say, I guess, or maybe auditory if it's something like siren.
Okay, so pick most common type of trauma, and then select. So then do you want to ask this question for a specific subject, or you want to ask for the population? Population?
Yes.
We'll have to generalize.
We'll have to generalize it, but we can start with one subject. Let's see how to do it for one subject, and then we can try to generalize it for the population. So I'll pick up a type of trauma, and I'll, once I've decided, I'll look at different types of trauma as maybe one type of trauma that I'm interested in, which is most common, which will help me find more subjects. I pick up that kind of trauma, and then I get some subject with that kind of trauma. Now what do? Now for this subject, you want to answer that which region of his brain gets activated when presented with the traumatic stimuli, or selectively activated, okay? So then what do?
So do we have to add more to the question, or is the question is the question satisfactory, or are we thinking of adding more to the question?
Well, let's start with this. Let's see where we get. Let's say we want to find selectively activated, okay?
So you're asking what would the next step be, right?
Yeah.
So I guess designing the type of experiment, as in what design to choose. So
This is event related.
What is event related?
Right.
I guess we would take.
Huh?
I thought we'd take block design.
Okay. What is block design? I have not covered, we have not talked about block design or event design. So what is it? You are going to have these people, and now you have fMRI scanner, and now you have to decide what to do. Present which stimulus?
The trauma stimulus.
Trauma stimulus. How do you know what is a trauma stimulus?
We first have to try to find actual people who don't have that trauma, and compare the results from them. Then
Okay.
we can actually try to give them.
Okay, select healthy subjects.
So that will be what our
Okay, I picked some healthy subjects also. Now what do?
We show both healthy subjects and then the same stimuli.
We show them stimuli. Okay, so there's a big assumption here, right?
Why it's
You sort of take 5 steps in one go, right? So what's the assumption here?
they're already in the fMRI machine.
No, that's okay. That's not a big step. I'm saying conceptual assumption or conceptual steps you're taking. 5 conceptual steps. Let's do one step at a time. So what do you mean by show them something? What will you show them?
The stimuli is visual in nature.
Yeah, so that's an assumption that their trauma can be induced by presentation of a visual stimuli. Maybe their trauma is not like that. Maybe it's an auditory stimuli. Maybe they have to get into that environment. Right? Somebody who has a claustrophobia, you may not be able to evoke claustrophobia, just by presenting a picture. Right? So, you want to get a stimuli that gives them a trauma, right? That sort of gets them to a traumatic kind of thing. How would you do that? So maybe you are going to pick subjects, again you're going to narrow down your experiment further. Not only common type of trauma, but you say that a common type of trauma which can be evoked by visual presentation.
So in homework 5, we had Peggy Palmer with visual neglect. So she was not able to pay attention to the left side of the image or whatever there was. So we can use that as a response here.
What do you mean by that?
So we saw that Peggy Palmer was not able to register what was happening on the left. So we can show a visual stimuli to Peggy Palmer or someone like her and an actual control subject and correlate how their brain activity is changing and be able to pinpoint it.
But how is it answering this question that you just selected, that which regions of the brain get selectively activated when presented with a traumatic stimuli? Yeah.
So can we use motor imagery to evoke, we ask that person to remind that traumatic situation that he or she had? And we can differentiate between that person in a normal relaxed situation and that heightened situation. And similar protocol would be followed for the other population, so that if there is a specific section which gets evoked for the traumatic stimuli, then it might be activated for that. So we might not need, it doesn't have to be specifically with visual or auditory. It can be, so the concept of motor imagery is you are activating that kind of sensation.
No, but how do you activate motor imagery?
We just ask that person.
How do you ask that person?
So we have to normalize that thing with the general population. So for one person, the trauma would be some kind of thing. Another person, trauma would be some kind.
Something
so
else.
We can't give same kind of visual or any kind of representation to every person.
Yeah.
But if we talk about ask that person to identify that what kind of, so we are selecting that most common type of trauma.
Yeah.
Maybe we ask them think about that kind of situation you are in, and if that person is actually having that trauma or felt that trauma, so some part of the region would be activated. That's my idea.
No, but see, again, how are you going to tell them to get into that situation? How are you going to do that? Imagine this patient or this person in the scanner. Now you want to evoke some kind of motor imagery in them. How will you evoke it? So you have to present some cue, and what will be the mode of presentation? It will be either an auditory cue, or a visual cue, or a tactile, or a taste or a smell. Right? One of those 5 senses it has to be. So which one are you proposing now?
I think it would be visual because...
Okay, visual. Okay, so visually what will you say? What will you present?
Jump scares, they usually scare movies.
Huh?
Jump scares, they usually scare movies. In the horror movies, it's suddenly some kind of...
Okay, so movies, movie clips. But then you are not evoking motor imagery anymore. Right? If you are presenting some movie clip that is a horror movie, then you are actually evoking something else. It may still evoke trauma, that's okay. But yeah. So here is the proposal that you take a clip. How long of a video you would need to be able to evoke that? Huh? Of a horror movie.
But that is
But
for
that
trauma.
is not related to.
You may invoke fear, but not trauma. That's a good point. Unless that person is traumatized by the same movie or a similar situation.
So that's the problem. Okay, we have to like generalize for the population. Like for different person will have different kinds of like
Yeah.
something will give trauma.
Okay, so let's simplify the problem. Let's say you want to find for single subject only. One subject only. That which area gets activated when that person is feeling the trauma or is sort of into that situation. So let's forget about, because yes, you are right. Different subjects may get, you may have a different stimuli to evoke the traumas in them. But let's start with one subject. So what do we do?
Maybe we'll first ask some questionnaires to understand what kind of trauma he or she has.
Okay, you ask them questionnaire. Yeah, what kind of trauma it is, how is it evoked, and how it can be evoked in the lab situation. How much is it safe to evoke? Right? You don't want them to feel
Ethical consideration.
Yeah, ethical considerations also, and you don't want them to feel overwhelmed as well. And let's say, you only select the subjects where the trauma can be evoked, or at least in some limited sense, trauma can be evoked by presentation of visual stimulus only. Right? So, single subject where the trauma can be evoked through presentation of visual stimulus. Okay. And let's say you ask the patient to give you the pictures, or you find the pictures, you make them sit in the lab, show them the pictures and ask them how they're feeling about it, and ask them to rate it. Are they feeling a little bit traumatized, more, and so on and so forth. So you based on this, you select a few pictures, right? So then you kind of, select. How many pictures you want to select? Okay, how many pictures? Just one should be enough? Why not?
Duration of attention. That might be there.
So if you only show one picture, the subject may get bored. It's no longer traumatic anymore.
It's
Make the evaluation robust.
Okay, so then how many?
I think it will be good also. Selecting multiple pictures might also be a downside, considering different pictures have different visual stimuli. Of course.
Yeah, different pictures can have different types of visual stimuli.
Visual
Right?
stimuli, and where maybe the effects that we are seeing is not related to the traumatic at all, unless of course we particularly look at only the amygdala and hippocampus, but then that would lead to the entire purpose. I'm not sure. Maybe one picture.
Let's say you find somebody who says, I get trauma feelings when I see pictures of snake. Snake.
That can also be
Huh?
That's phobia. If they have had a traumatic incident.
Maybe they've had a traumatic incident in the past. Or let's say a particular kind of snake, right? Let's say cobra, king cobra. Right? There was some news in king cobra in Metro or something. In Indian Rail, some news I was watching. So let's say, some kind of a cobra snake, but not with other type of snakes, let's say. Maybe they have some kind of association with that kind of snake. Okay, but then how many pictures you want for that type of snake?
8.
Huh? 8. Okay, so let's say 8 to 10. But why 8 to 10 pictures?
I think you would want different pictures of the snake, but throughout those pictures you would want to control for the visual elements, the color, saturation.
Hmm. Okay. So do you need other snakes also? Pictures of other types of snakes?
We should include some other pictures like which do not invoke the person's trauma, because we want to see kind of a reference.
Okay. And what type of pictures?
Fixation kind of pictures.
That do not evoke trauma, right?
Yeah.
Okay, so one candidate is fixation. What do you mean by fixation? A white screen with a black cross in the center, or a black screen with a white cross in the center. That's one candidate. Okay. Now what? Okay, so we have a fixation, we have 10 pictures of snakes that, if the person sees, he or she may get traumatized inside the scanner upon seeing them. Now what do you do? You put them inside the scanner, you start the scanner, start collecting the bold fMRI data, but what is there on the screen? You see.
Correct the decide on the interval.
What interval?
Order matters of
Order.
Huh? Order?
We can place a few images of the snakes which are not causing trauma first, like 2 or 3, and then we can place 2 images of snakes which do cause trauma.
But here we said we'll just do fixation.
Fixation.
Okay. So then what do we do?
Decide on the interval, as in how many seconds of
Okay.
fixation and how many seconds.
So decide
20 seconds.
why 20 seconds?
Because that's what we saw in other experiments.
What is that experiment? That's not a reason good enough.
So it could be like
Because it is a trauma and it
Huh?
would be of less time.
Could we have less time, but how much time? Yeah, you were saying something.
fMRI are normally 152 milliseconds, if I'm not wrong.
What, 150, 200 seconds?
Like the total time is being recorded.
Total time can be up to an hour. Few minutes to an hour. Half an hour, easily you can do. Yeah, so what will you do?
Shouldn't we first take a baseline first?
How would you take the baseline?
Like with a 30-second fixation.
Okay, so you start with 30-second fixation. Okay.
So we know the baseline that when no stimulus is given, how the brain is actually reacting.
Okay.
And based on that, we then give a 15 or 20 seconds image, 20-second image of a cobra, then another 10-second of fixation.
Okay, so 20-second cobra.
Yes, sir. And after that, a 10-second fixation.
But why? Why 20-second? Why 30-second? Why 10-second?
So I have a question over here. Like we have, let's say, 30 seconds of fixation and suddenly we show a picture of the cobra. And we are assuming the person has traumatic fear of cobra. And the rise in whatever bone signals we are seeing, we are assuming it's because of the trauma or because of the fear. But
No. It's because of the stimulus.
okay.
Whatever changes in the bold signal happens, it's because of stimulus. Okay. Yeah.
the bold signal, the HRF rises with there is a delay of 2 to 5 seconds, and because of that, I think we need this duration.
Okay, so if you present a stimulus that is getting some brain areas to get activated, it's going to say take up to 5 seconds for that area to actually become activated. Right? So if you remove the stimulus too soon, and then you present another stimulus which activates a different area, then they might get jumbled up. At least for now. That's sort of. So you want to at least present it for 5 seconds, maybe a little longer, maybe 10 seconds, so that whatever changes in the bold signal happens, that stabilizes. You can get to that change and you can actually measure that change. So maybe 10 seconds is a good number, right? Maybe 20 seconds is a good number. But certainly not less than 5 seconds. No? Because then, this can get confusing. Because of the hemodynamic response delay. Okay? So we start with the 30-second fixation, then 20-second cobra. And also, why not 1 minute? That's on the lower side, we said it should be more than 5 seconds, maybe 10 to 20 to 30. But why not make it 100 seconds, maybe 200 seconds?
Might make the patient panic.
Huh?
Might make the patient panic.
Might make the patient panic, okay, that's a very fair point. You don't want the patient to be in that state for long, okay. There's another reason, I'll talk about it, it's called fMRI trends. In the fMRI data, you get some, trends because of scanner artifacts, not because of stimulus. So if you present it for too long, then the change in the signal, you cannot distinguish the bold signal from the scanner noise because of the trends. So we'll talk about that. So maybe up to 30 seconds, up to a minute is maximum, but you don't want to go more than that because otherwise it becomes difficult to separate the noise from the stimulus. And of course, you don't want the patient to panic inside the scanner, right? So that is also another good reason to limit the stimulus presentation. Okay? And are we done? 30-second fixation, 20-second cobra, and 10-second fixation. That's, in about a minute your experiment is done. Now you can analyze the data. No? Yes.
We have to make sure that the brain area that is evoked by this 20 seconds of cobra is not actually evoked by any other image.
Okay.
In case of 20 seconds of cobra, then for 20 seconds we give some other stimulus.
Okay, so what other stimulus you would want to give?
Like a field.
Okay, so let's come back to our question, right? So I think let's skip this part selectively for now. Here we are saying selectively gets activated. Let's say we want to get started with only activated, okay? Which part of the brain get activated when presented with this traumatic stimuli, okay? So then is this enough or do we need to do something else?
We need more sessions with the patient because of daytime effect. Only from one day.
But for one session, this 1 minute scan is good enough?
We need more batches of data.
But why?
This batch represents just one instance of that particular.
One trial, as they call it, right? One trial. But why do you need more trials?
Normalize the data. Maybe there might be some effects before the experiment and something after the experiment.
That you have taken, right? You have taken a fixation. And then it's cobra.
So that we can be sure that it is the stimuli that, it is a specific stimuli that created that response.
That want to show, yes, absolutely want to. And what could go wrong? Let's say you do this, you analyze the data, you find this brain area, in this period, this brain area had higher activity.
It could be at random.
It could be because of random noise. So, may corrupt your observations, right? So what do you do then? So one trial is not enough because each trial has, we talked about it, right? The scanner noise and other kind of noise is very large, right? The bold signal is very, very small. So if you just do one presentation, you'll see, half the brain lighting up because, it's kind of noise. You can do it with your assignment 1. How many blocks it had? 3 blocks it had, right? 3 presentations. You can just look at one presentation and what happens, right? What areas you see getting activated, right? You have a baseline which is a fixation, and then you have a checkerboard. You can actually see that. Okay, so how do we take care of the random noise?
We take multiple trials.
So you do multiple trials. Okay, how many trials? So how many trials you need actually depends on the statistics, right? Depends on how much is the strength of the signal, how much is the noise. And once you do the stats, you can actually figure out the number of trials that you need. Because you need to see how much is the signal change, related to that how much is the noise. And then how many trials it will kind of cancel out. But let's say we do 20 trials. Also depends on how much time the patient can stay in the scanner. But let's say for some reason we decide 20 trials. We do the analysis, we say 20 trials should be sufficient. Okay. Now what can go wrong? So what I have is start with a 30-second fixation. Then I present 20 trials of 20-second cobra image and 10-second fixation. Right? And I have only taken one cobra image as of now. And I'm then presenting the same image over and over again, right? 10 times, 20 times.
Can get habituated to the trauma might subside. You might get used to
Desensitized.
He may get desensitized, he may get bored. Right? So then how do we fix that?
Randomize the trials.
Huh?
Randomize the trials.
What do you mean by randomize the trial?
So, say we have 5 blocks of these like first 20 seconds of cobra, 20 seconds of fixation.
Mm-hmm.
Like for the we randomize for the first 6 session, we do like 20 seconds of cobra and 10 seconds of fixation. Then after a quiet fixation, there is nothing in there, just X mark in the screen.
That is fixation, no?
But for the 10 seconds, like then we do it again. After 10 seconds, we do another 10 seconds of fixation. Then with the give that cobra stimulation, and again for the 10 seconds of like you will randomize both. Because there's only 2 options available. Like usually in the whatever the experimental protocol we do, like we have a number of different things to like randomize the problem. But for the 2, I don't know. Like, but we have to somehow randomize it so that they don't get habituated or they don't like it.
No, but you have to be a little more careful, no? What is the problem that you're trying to solve? Right? There are multiple issues. One issue that you talk about is that he may get, the trauma may not evoke anymore because, he has already seen that, so many times. So how do you solve that problem?
Present new pictures, I guess. That's the only thing.
Present new pictures, Okay.
That's the only thing we can think that we can do. But otherwise, desensitization is not avoidable.
Okay. Right? So this is a trial. This is what we call a trial. Okay. So the patient may get habituated, may not evoke trauma after 3-4 trials. What is the solution?
So we could give a resting time for like after 5 trials.
So present different pictures, no? In different trials. Okay?
But still that
No, you're not. I mean, you just said we started with sun, but now you're doing different pictures. But there may be different types of habituation, right? That you know that whenever the fixation goes, a cobra image will come, right? Maybe different cobras every time, but still a cobra. So still some kind of habituation may happen. So how do you solve for this?
The only way is to find different people. I mean,
We are looking at one person right now. Because all the pictures have cobras.
like, I guess like Rohit said, we can measure time duration between successive trials.
Yeah, time duration between successive trials, but that will not, why will that solve the habituation problem?
They are not looking at it for a continuous period of time, right? So when you're giving a break, like it almost as a new.
So what are you suggesting? Instead of 20 seconds, we present it only for 5 seconds?
No. So there are 20 trials, right? So after every 5 trials, there will be like a resting period for 2 minutes with just the fixation cross show.
But fixation, see, basically you are saying increase the duration of one of the fixation blocks.
No, I mean then some here because the second image is a fixation cross.
Yeah.
But if it was some other image, we could show a fixation cross for that period of time.
But you are deciding the protocol. So you want to show other images?
You could show a non-snake image
Okay.
because in the end we actually want what causes the trauma, not the shape of color.
Yeah. Okay. So now, what are you proposing? Your protocol becomes a stimulus presentation there. So,
there will be a 30-second fixation,
30-second fixation,
20-second image of cobra,
20-second image,
and 10 seconds or something like that again fixation.
10 seconds of fixation,
And then a 20-second image of a non-cobra.
So let's say this is a trial, okay, and we repeat the trial. But now there are two types of trial, right? There are trial with cobra images, and then some wildlife. Right? So there are two types of trials. So you show either a cobra image or a wildlife picture. And then you do a fixation, then again show the image, then again the fixation and so on. Now what do? You do alternate, you repeat one after the other.
No, then they will be expecting.
Then they will be expecting. And if they expect, then the brain areas may start getting firing even before the stimulus is presented because they're already expecting something. So what is the problem now? Expectancy. So let's call this as trial 1 and this is trial 2. So expectancy may happen if trial 1 and trial 2 are presented alternately. Okay, one after the other. And there's one more cause for expectancy. even when you do not present them one after the other, there is another cause for expectancy related to what you were saying. What is that cause of expectancy?
सर अगर हमारे पहले कहा था कि हम कोबरा के ट्रॉमा का टेस्ट लेंगे तो
उनको कुछ नहीं बता रहे, उनको कह रहे हैं वहाँ लेटो और पिक्चरों को देखो और जब क्रॉस आएगा तो क्रॉस को देखो, आंखें बंद मत करना, टेंशन नहीं रखना, इतना ही बोलेंगे. हाँ, व्हाट? हाँ, क्वेश्चन वी टुक, दैट्स ओके. हाँ, एथिकल कंस्ट्रेंट में कोबरा को दिखाएंगे, बाकी और भी दिखाएंगे. शो देम अदर पिक्चर्स आल्सो. वाइल्ड लाइफ की पिक्चर दिखाएंगे, एनिमल्स की दिखाएंगे, स्नेक्स की दिखाएंगे. बट या there is one cause of expectancy when you present these cobra and wildlife images alternatively in one trial. So, for example, you can do, you can have something like, trial 1, then you do trial 2, then you do trial 1, then you do trial 2, right? So, this is cobra, this is wildlife, this is cobra, this is wildlife, right? So, then they will begin to expect. After they've seen the cobra image, fixation comes, and next time they will expect a wildlife image. So, that is one cause of expectancy. So, instead of this, if I randomize whether to present cobra or wildlife, I just toss a coin and then do it, then this expectancy will be gone. Because they don't know in the next block what is going to come. But there is yet another cause of expectancy.
So, in a trial, first the picture is shown and then a fixation cross. So they know that after a picture, a fixation cross will be shown.
Yeah, and not only that, the duration of the picture is fixed, right? 10 seconds of picture, 20 seconds of expectation. So they know exactly when the picture is going to go away and the fixation is going to come. So this fixed duration, right, may also cause expectancy. So what do we do? So randomize this also. I randomize the times, then I'm done. I solve the problem of expectancy.
If we randomize that duration as well, won't there be another variable that can affect the result as well?
Which one?
This one. Okay. Like, because we are randomizing the trial, right? And again, the duration is also randomized. So some time will come like in 5 seconds, 6 seconds. Yes, we will be like expectancy won't be there. But won't there be, when I am going to analyze the data, won't it be much more difficult to like normalize or generalize the data?
Yes, so that's a very good point. That's an excellent point. If you are changing these durations, then how do you analyze the data? Because you are presenting the stimulus, you can record when you are presenting with stimulus, and that record has to be there. But using that record, you cannot just take an average, because anyway the bold signal is this messy signal, which has a lot of noise and it's convolved with HRF and all of that. So how do we analyze that data if we actually make these times so variable? So I'll come to that when we talk about the general linear model. It actually solves that problem. It tells you how to analyze this data. Okay. So fine. So if you do this, and then if you can somehow find that, which brain areas become more active. So now what is the question that we have to answer?
Which brain areas activate?
So, one doubt. If we randomize the time for each image and do multiple trials, how can we be sure that noise is actually discarded from it? Because as I believe the duration should be exact We actually want the trials to decrease this noise.
Yeah, so what you have is I'm going to represent trial 0 or trial 1 by blue and trial 2 by green. Right? So when you look at this, you may have a picture like this, right? Different trials presented at different times, different types of trials, right? And then when it is 0, it also represents fixation cross, right? So this means fixation cross, this means fixation. Okay. So let's bifurcate this into two pictures. One would be something like this, the blue one. And let's call this S1T. Right? This is a trial 1 presentation. And the other picture we can bifurcate as the green one. Something like this, right? And let's call this S2T. Okay? And this is 0, this is 0, this is 1, this is 0, this is 1, okay? Okay, so now what is the analysis that you want to do? So you collected the bold data, and now you want to find activated regions, right? Which brain regions are activated when S1, stimulus 1 is presented, right? Okay.
And subtracted from the wildlife one?
Subtracted from
Wildlife.
S2, right? Second. We are doing selectively activated right now, so we are just doing activated. So then, what do we do? So if a brain region is activated when the cobra image was presented, then how is their bold response likely to be? So of a brain voxel, we use a brain, each point is a voxel in the data. Now in a 2D image you have a pixel, because a 2D, in fMRI we have a 3D picture, so that's why it's called a volume, and that's why we call it a voxel. And you can think of this data as, multiple voxels, right? Each voxel has its own time series. So what is the expected bold signal for an activated voxel? Right? Let's call it a voxel number I. And let's represent that as YIT. So YIT is the expected bold response. YI ideal. What will it be?
S1T convoluted with H.
Yeah, X1T convolved with H of T. And that's what we would get. But times some beta I. Beta I is a scalar. Because see, different voxels may get activated by different magnitudes. Some voxels may have very small activity, some voxels may show a very large activity. So S1T is 0 and 1. And HT is, we are again, it's a standard function. So to represent the variability across different voxels in the response, we are also doing a beta I, and this beta I is essentially represents the strength of activation. Okay? How strongly it is activated. And if a voxel is not activated, then we can say that its corresponding beta I is 0. Okay. So yeah.
Sir, and how do we find strength of activation?
Yeah. So how do we find strength of activation? Good question. Right, so now actual activity would get some noise added. And this noise would be scanner noise, physiological noise, and psychological noise. All three, we're just modeling it like that. Right? What's our goal? So the general linear model analysis that I was talking about is given YIT and this SJTs, right? All the stimulus time courses. Find beta I. And that's what it does. It actually gives you beta I. And whatever beta I you find, it may be influenced by this noise that you see here. Because you don't know this number, no, you only know this plus noise. So if noise is very large, this may influence this. So we want to find it in a robust manner, in a statistically robust manner. So that's what we will cover in the subsequent classes. Okay? So yeah. So this will tell me if somebody can, from this data can give me the beta I's, then I'll just check whether it is positive or not. And for the voxels where the beta I is positive, I can say they are getting activated when the response is there. And when beta is 0, I say they don't get activated. And when beta is negative, I say that particular voxel gets deactivated when the stimulus is presented. So now how do I look at selective activations? Now I want to look at selective activations, area that get activated only if, that kind of stimulus is presented.
Subtract this the result from S2.
Yeah, but what is S2 now? You have to be carefully designing S2. Right? So we do these two types of trials and we have, this kind of a design. But what to present during S2? Right, so we remember Nancy's talk. She did exactly that, right? So, she could not conclude just by looking at faces alone, if you get some areas activity, that it is selectively responding to faces. It could be just responding to the color, responding to the shape, responding to many other things. Same problem that we see here. So, yeah. So now we want to find out brain areas that are selectively responding to the trauma stimuli. And for this particular subject, I know that, snake or cobra snakes are the cause of traumas. Right? So now what kind of, how would you, what would be S2 now?
So
We show other kinds of snakes.
Maybe we'll show other kinds of snakes. But those images you should also match so that the color and the composition and other statistical properties, the luminosity, the contrast, the quality, all of them are very similar. So that we can be sure that the differences that you are seeing are not because of those image attributes, they are actually because of snake.
This actually creates one more problem. There's a possibility of classical conditioning happening if we associate a traumatic stimuli with non-traumatic one.
But how are you associating it?
Especially considering if we are trying to make it very similar to.
No, but how are you
but
associating
they
it?
want to get it conditioned.
No, but they are getting the fear response is triggered due to the traumatic stimuli, and then immediately after that we are showing it non-traumatic stimuli.
No. What are you saying?
Getting a fixation cross. After which the non-traumatic is.
Oh yes, so there is going to be time division.
And then that need not be non-traumatic always, right? It could be traumatic again because you're randomizing. And the duration is also randomized. And the next stimuli may be traumatic, may be non-traumatic, because again that is also randomized.
So along with other snakes, should also include like stuff like patterns, the snakes.
Maybe like rope or other kind of patterns.
Yes,
Yes.
and
Yes.
also drawings of maybe snake like baby patterns. Something like that.
Yeah. So S1 could be snakes. Then let's say S2 is other snakes, non-cobra. And then maybe you can also have a third type of stimuli where you have shapes. Lines, scales, ropes, etc. Right? So those kind of pictures you can show. Similar background, in the similar background. Okay? Fine. So you collect this data. Now what?
Same design.
Hmm? Okay. So what do you talk about? So we already talked about these things. Let's quickly go over it, right? So the bold signal is impacted by neuronal firing, physiological and psychological state of the subject, and noise. Right? So there's a lot of noise that happens, right? Statistical, physiological, and psychological considerations. We already talked about, expectancy, and because of that we are doing the randomization. We didn't talk about physiological considerations, but maybe there is a change in the breath, maybe there is a change in the heart rate. We have to normalize that somehow. And we talked about repetition, right? We have to repeat it multiple times to take care of the statistical errors. We're talking about compare the task of interest with some control tasks, right? And a simple baseline task, like a fixation, right? And then repeat this as much as possible so that you can take care of the statistics. Okay? And yes, repetition is one of the key things, and the other thing is comparison between two tasks. Right? A baseline task and a task of interest. And in this case, task of interest is a cobra picture, and there can be multiple baselines that we can look at here, like snake pictures which are not cobra or similar objects. Okay. We also talked about expectancy, how to deal with the expectancy. Another thing is adaptation. That subject may get bored, it's been in this panel for so long, same thing is happening over and over again. And we are changing the pictures. But even within that 20-second or 10-second picture presentation, you need not have one picture. You can actually show multiple pictures, one after the other. So that will also take care of a little bit of interest of the subject. And they will not get that bored and adapted. Okay. So this is one type of design that we did, and this is what is called a block design. In a block design, each block is like 10 to 20 to 30 seconds, and within that block, we are presenting the same type of stimuli. So in the cobra block, you are only presenting cobra images. There may be a single cobra image in that block of 20 seconds, or there will be multiple images. So you can show 2 seconds each image, right? But all of them are cobra images. But you can also do a paradigm which is also called event paradigm. Right? So here is an example of a block paradigm. You have condition A for 20 seconds, then condition B for 20 seconds. And within condition A, you can have multiple stimulus of the same condition. Or you can have the same stimulus also. And how do we analyze the block design? You have these blocks, you have a hemodynamic response function, you convolve with HRF, you get a pattern like this, and this is your predicted bold response. And then you correlate it with the data. But you can also do event-related design. So what you can do is, and condition A and condition B, you can present that stimulus for a very short duration, less than a second, and then you change. So here, for example, you presented, so this is condition B, which is like a non-cobra snake, and then a fixation in between, and then a cobra image, but for a very short duration. And then you keep doing that.
I have a question over here.
Yeah.
So you said like the response, the stimulus response product you showed
Yeah.
has some kind of lag and it
Yeah.
So if we show images for this short interval, like will that reach the peak or how
Yeah.
will it show?
So again, so look at this here. So you have this is, let's say, cobra image presentation, right? So, each of these presentation is like half a second or a second, very, very short duration, but they are happening multiple. Then what you do is convolute hemodynamic response function. And you will get a signal that you see like this. Now what you can also do is, non-cobra. And then you'll have these, presentations for the non-cobra. Again, you convolute with hemodynamic response function, which is the same. And this will give you another time series, which will be different from this one, from the first one. So this gives you the expected bold response for the non-cobra. Then you are going to look at the difference and compare where the signal, which voxel, cobra images is more correlated. And you can also do a mixed design. Okay. What's a mixed design? Do I have a picture here? So you can have a block of a particular type, and within the block you can have events. So they are called mixed designs. So for example, you can present pictures of cobra snakes and non-cobra snakes, and you can look at how the meditators behave with respect to non-meditators. Or rather, how do meditators behave when the stimulus is presenting while they are meditating versus while they are not meditating. So you can have one block where they are meditating, and then you present these two types of images, and then another block where they are not meditating and present these two types of images. So now you have mixed blocked and event design. So each block represents the state of the person, and the event represents the stimulus presented. Okay. So we talked about repetition. Yeah, we talked about repetitions. We talked about designing contrasts, and then handling expectancy and other physiological effects. And yeah, that's an important point we did not talk about. Subject motion. If a subject moves, the data becomes corrupt because it's very sensitive data to motion. And I will explain why in the subsequent lectures. So whatever design you do, you have to make sure that subject can do it without moving their heads, even for a millimeter. So if there's some design that requires them to breathe heavily or require them to move their shoulders or hands, that may introduce motion in the hand. And this can corrupt the data.
Can we expect even the slightest motion to correct the data, or is it?
Yeah, generally advisable to minimize it.
But considering how long they are going to be in the machine, wouldn't it be?
It's a challenge, yeah. It's a challenge. What we typically do is wherever subjects have large motion, we discard that data.
Oh.
And collect again for new subjects. It's hard, yeah. Okay. Yeah. So now you have two conditions, A and B, cobra and non-cobra. And then you want to find out. So there are different types of analysis you can do. So the simplest is subtraction. What does subtraction mean?
Subtract, reduce one thing by the other.
Yeah, so if I get, S1t convolved with h of t, right? That's for the cobra. And I subtract it from S2t, convolved with h of t. And then see whether this is no, actually not this. No, not this. So I look at I is the voxel number, right? S1t convolved with h of t times beta I1 plus S2t convolved with h of t times beta I2 plus noise. Okay? So look at it carefully. This is my voxel I at time t, and this signal is my first signal, right, with my first stimulus, which is cobra images, with the strength of beta I1. And second is the snake images with the strength of beta I2. And then the noise, the random noise. So I model this and I have values of beta I1 and beta I2. Right? And my baseline condition is a fixation. So if I look at a voxel in my visual cortex, in the subject's visual cortex, they are going to be activated when the cobra image is presented. They will also get activated when other snake image is presented. So for those voxels, beta I1 and beta I2 would be very comparable, very similar. Both will be positive and they will be very similar.
Sir, but at a particular time, wouldn't there be only one of those two stimuli?
Yeah.
So then why are we adding both here?
Because
Are we telling that okay, one of these is going to be zero, so this is just a general equation of the
this is a general equation, yes. At any time t, you look at the presence of this stimuli S1 and you look at the presence of S2, convolve with hemodynamic, look at the strength of activations, add noise, and that's your final activation. Now with this, I can get beta I1 and beta I2. And for this voxel, if both of them are positive, both of them are comparable, then I can say that both the voxels are activated. I mean, this voxel is activated for cobra as well as non-cobra images. But if I can get that, if I can somehow conclude that beta I1 is much greater than beta I2, right? What does it mean? That this voxel is getting more activated to the cobra images as compared to the non-cobra images. This is the same as saying that beta I1 minus beta I2 is greater than 0. So then I can say it is selectively more responsive to stimulus 1 as compared to stimulus 2. Okay? And this is what is called the subtraction design. I look at the effect size and I subtract one from the other and I want to find whether it is greater than 0 or not. It could be because of trauma, that this voxel is getting more activated because this image is invoking trauma, other image is not. It could be because of other features of the snake. But I have designed block B, I have also designed block C, which is like other snake-like objects. So I can look at this is greater than 0, and let's say there's another one, S3t convolved with h of t times beta I3. So I can look at beta I1 minus beta I3 is also greater than 0. So if both these conditions are satisfied, this means it's more responsive to the cobra images as compared to snake images, and it is also more responsive to cobra images as compared to snake-like objects. Then I can say that it is selectively responsive. So now I would suggest you go read the paper again. Okay? Now you will be able to understand all of these, right? Subtraction, factorial design, parametric effects, and conjunction analysis. And
I didn't understand why we are adding S1T and S2T.
do we have time? Because I'm out of time. So do you guys have classes after this, or you are available? Available. Okay, so fine. We can just take 5 more minutes to Okay. So let's look at this, right? So this is your S1T. Right? And let's say this is your S2T. Okay. So I convolve with hemodynamic response function, I'll get a response of something like this, right? So that's my expected board response for a voxel which is getting activated by the stimulus 1. But if a voxel is also getting activated by stimulus 2, then for stimulus 2 it will respond like, right? Something like this. So when I am presenting both stimulus 1 and stimulus 2, my expected response for a voxel that gets activated both for stimulus 1 and stimulus 2 would be the sum of these two. Okay. You don't agree. That's a very good point. Because why will it be a sum of these two? It will be a sum of these two because of an assumption we make in the analysis, which is called the linear and time invariance. And because of that assumption, I say that if two stimulus are presented and they are giving the same response, they are giving some responses, then if I present them together, if I add them up, I'll get the response which is also adding that up. So therefore, my final response would be something like this. It's something like this. I'm sort of, excuse my drawing. And so therefore, my y of t would be S1t convolved with HRF times the strength of activation because of the first stimulus, and S2t convolved with hemodynamic response function and strength of activation because of the second stimulus plus the noise. Does it make sense?
Sir, but at time t, S2 is not being presented, right? So, I mean...
Yeah, so let's look at one particular time t here. And here we see S2 is not being presented, S1 is being presented. So this signal is 0, but this signal is getting added. Right? But at this particular time, this one is 0 and this one is positive. So therefore, as a general function of time, because this one yt I have to get for all values of time. So how do I write it as a signal? It's like a signal, so I have to get its value at all values in time. So therefore I just combine the two and then I'll get it for all values of time. So if I write it like this, then from this I can actually get the values of beta 1 and beta 2s. And there are methods to solve it that we'll discuss later in the course.
So there is no actual subtraction going on here?
Actual subtraction is going for the betas, not for the signals. You will do some analysis on top of this and we'll get the beta values and we'll subtract the beta values.
But if S2 is 0 at time t, then wouldn't beta I2 also be 0?
No, beta is a constant, it's not changing with time. Beta 2 is constant. So S1t is changing with time, h of t is changing with time. So if you look at S1t changes with time, t changes with time, but beta 1 is just a constant, it's a scalar. Similarly, these two are signals, but this is a scalar. So this doesn't change it.
That would also be an assumption.
Yeah, this is what is called the linear and time invariant assumptions. We'll talk about it in the next class. And that is one of the fundamental assumptions which is wrong. But, it's still good enough to be able to get to a lot of good, interesting things. So stop here.